Thank you everyone for joining today. My name is Diane Bossenjack. I will be your support and contact during this webinar. So as a quick little reminder and some housekeeping here, this webinar is being recorded for archival purposes. We will be posting it again on the BCoN website and on the YouTube page, and I will let you all know where that will be. So if you have any questions or issues during this webinar, please feel free to use the chat feature below, and someone, specifically me, will assist you. There will also be a Q&A session at the end of the presentation. Please use the Q&A feature that is located on the bottom of your screen to type in your question. Feel free to type it in at any point in time, and the presenter will go ahead and we will address those at the end. So I'm going to go ahead and get started for this. So again, thank you for joining. I'm going to go ahead and hand it over to Dr. Andrew Bentley from at the Biodiversity Institute at the University of Kansas. Go ahead, Dr. Bentley.
Hi, good afternoon, everybody. Mr. Bentley, not Dr. Bentley, but hopefully you can all see my screen. So a lot of you know me, but for those who don't, my name is Andrew Bentley. I'm the ichthyology collection manager at the University of Kansas at the Biodiversity Institute. I'm also the bioinformatics manager and do some work for Specify. I am also one of the co-PIs on the Biodiversity Collections Network grant, the BCoN grant, and also a past president of Spinach. So what I'd like to do today is talk to you a little bit about data integration and attribution. A couple of things have been going on recently about this in terms of some of the BCoN things that we've been initiating. And so the first part of the presentation, some of you may have seen. It's my sort of soapbox presentation that I've been giving at various conferences and such recently. But then the end of the presentation is more about a needs assessment workshop that we held recently at KU to look further into this and try and come up with some solutions to this problem. There we go. So back in 2012, NSF funded a group of people to get together to come up with this NIBA plan, the Network Integrated Biocollections Alliance Plan, which had a number of goals outlined in the plan to do with digitization and various other aspects of collections. One of those goals was goal number 2, which was advance the engineering of the US biodiversity collections cyber infrastructure, which has very great bearing on the kinds of things that we are talking about now in terms of integration and attribution. As part of that goal 2, there were a number of sub-goals that were highlighted, and I'll let you read through those. I'm not going to go through all of them, but 2.5 is the one that's of great significance here, is anticipate the future of biodiversity specimen data integration. And so, you know, this got me to thinking that maybe this is something that we should look at in more detail. More recently, the GBIF has put out this Global Biodiversity Informatics Outlook document, which focuses on very similar issues, harnessing the immense power of information technology and an open data culture. It too had a number of sub-focus areas that they were looking at, and again, a number of those align with the kinds of things that we are talking about here in terms of integration and attribution. So recently, we've been making great strides in various areas associated with digitization. IDIG-Bio, ADBC, and the TCNs and collections in general have been digitizing more specimens and publishing more data through the various data aggregators that are out there. Collection management software has been facilitating this publication through the expanded data models that they are now integrating into these systems, and also the integration with IPT and Darwin Core to facilitate publication of that information to the outside world. There's a number of groups working on best practices, protocols, and workflows that are being disseminated through various workshops and webinars and wikis and various other outlets. And BCoN and IDIG-Bio and Spinach and NSCA and others are involved in galvanizing the community around this common cause of digitization and integration. So there are also these folks and other folks who are also involved in training the next generation of collections personnel, looking at undergraduate training and graduate training and all of those kinds of things. what I see as being the huge missing piece here is the integration of these data components into a seamless data pipeline that can make this data more available to the ever-increasing and varied external user community that is now using this information for all sorts of different purposes. We need a flow of data from collection of data in the field to the collection, to the aggregators, to repackages, to research publications, and to external users, with as little human intervention or impediments as possible. All of these individual components are obviously essential cogs in the functioning of this collective cooperative data pipeline. And it also would facilitate the interoperability of our data, integrating it and scaling it with other sources of data outside of our immediate community. The attribution portion of this is we obviously need flow of information in the reverse direction, back to the collections, to allow for collections advocacy through the attribution of all of these products and show of use and need of these collections. And despite the newfound spotlight on the value of collections in publication and popular and social media, collections continue to come under pressure. They struggle to show relevance and utility and are hampered by diminished funding and staffing. There's a number of metrics that collections use to show use and activity. Some of these are through publication, citation, collection visits, etc., as well as data usage from portals and aggregators to show the relevance of these collections and how important and useful they can really be. Managing this attribution is becoming exceedingly difficult for collections to do because of the proliferation of end users now using our data from numerous different spheres. And also the data becoming more ubiquitously available from multiple sources. So if we look at the sort of landscape of what our community looks like in the universe, this is a diagram that I drew up some time ago, looking at the various levels of sort of interaction that go on in our community. It obviously starts out with the data and specimens being collected in the field and then being databased into these collections, no matter what kind of collection they are: federal collections, university collections, small collections, tissue collections, living collections, whatever the case may be. Thereafter, these collections are then obviously databased into some sort of databasing software. There are numerous different kinds of software out there that people are using for collections, and this is the primary vehicle for making this digitized information available to the outside world, not only for research but for other people to use. These databases then feed into aggregators thanks to Darwin Core and IPT, to make this data then available to the larger research community and allow people to be able to search multiple collections all at the same time. Along with these aggregators, there are then also repositories of information. I call them repositories. They are systems that provide specimen information as a sort of secondary result of their initial goal. Their initial goal is not to provide specimen information, but as a side effect of that, they do. For instance, places like GenBank, where their primary purpose is to support the housing of GenBank sequences, but there are also connections to voucher material and tissue material that are associated with those kinds of things. Then you have this large and ever-increasing external user community. Obviously, the bulk of that is coming from taxonomic and systematic research, which is the main reason why collections are being used. But there is this ever-burgeoning group of external users who are starting to use specimens and data associated with collections to answer all sorts of questions. Bigger questions like climate change and invasive species and those kinds of things, but also all sorts of other things like environmental impact assessment, et cetera, et cetera. And so, trying to get a handle on who all of these people are that are using our data is becoming increasingly difficult and more laborious. And so we obviously need this flow of information going outwards from the collections outwards to the research community and other end users who are using our data. But we also need a flow of data in the other direction in order to allow the collections to then take all of this data that's being generated by research and various other things and use that as an advocacy mechanism to then be able to advocate for these collections and show their use and utility. So this is another diagram that I drew up some time ago just to explain the sort of landscape of a collection and how all of these different components fit in. So this is looking at my ichthyology collection here at KU. So we obviously have a voucher record that has a GUID associated with it. And that voucher record also has a tissue record associated with it, a tissue that's associated to the voucher that is then used in research. Along with that, we have images of the specimens. We may have x-rays of the specimens and all sorts of other digital media that's associated with the records themselves. There are also field notes associated with that record that may give additional information that potentially may not be found in the database itself. But increasingly, we are now seeing products created by the research community that are not being transferred back to the collections. So images and various other resources that are being created in publication, or images that may not even make it through to publication, that are being taken of specimens that don't necessarily get repatriated back to the collections themselves. There are also things like the new OVERT grant, MorphoSource, and various other CT scans that are now being produced and are out there in the world and are being connected back to the vouchers or tissues that are contained in the collections themselves. So we also have our own web access portal where people can go and search our collections, as well as porting our material out through IPT. We also have our own web access portal that allows people to search the collection directly. So you have these numerous different pathways in which people are using these collections. Obviously, the main avenue of getting our data out into the outside world is through the IPT client. This is the IPT client for KU that has all of our collections involved in it. And those are then ported out to all of the different aggregators. So our fish information is ported out to FishNet. It's also ported out to VertNet, and there again you can see the GUID that's associated with that record, all of the associated references and all of the associated sequences that are associated with that record that are also being ported out through the aggregators themselves. So we're starting to make some of these connections to various disparate pieces of data associated with the records themselves. Again, there's the same scenario in GBIF. You can see the occurrence ID, the associated sequences, and the associated references, and the same thing in IDIC-Bio. So there are some of these connections that are starting to be made. Then we obviously have a whole bunch of GenBank sequences that are created associated with a particular tissue record in our database. And all of those GenBank sequences hopefully are then connected back to the actual record themselves in our collection. So here again, you have a link to the actual publication that's associated with those GenBank sequences. And also on the right-hand side, we have a link out to the actual specimen record itself in Specify 7. So that folks can actually go back and see the actual record itself to verify the identification, to see if any data has changed since that record was submitted. And there are a number of cases where IDs have been changed, various other parts of the record have been changed since they were lodged with GenBank. Then we also have a number of publications that are obviously citing material from our collections, and this is a minefield in and of itself. In one of the publications, there is actually a listing of all the GenBank sequences within the publication itself, showing all of the tissue sources, so all of the KU numbers that are associated with the tissue records that go back to our collection. In the other reference, unfortunately, all of that material is in the supporting information. And so it's very difficult for us to go in and troll that information and try and find reference to our materials. But all of this stuff is being done by hand. So all of this stuff is being generated by me by going out and trolling GenBank and trolling the reference list to try and find information associated with our collections. And it's hundreds and hundreds of hours worth of effort on my part to try and make these inferences and make these connections between these disparate pieces of data. And so this is another thing that I've done, is creating a Google Scholar profile for our collection, indicating all of the citations and all of the reference works associated with our collection. And that H-index of 79 is a pretty good indication of how important our collections are to the research community. But then there's also this great unknown. There's this great unknown of who else is using our data and what are they using it for. And that is even more difficult to get a handle on. So we have a number of tissues and a number of lots associated with our collection. We have lots of images and field notebook pages, about 14,000 GenBank sequences that I've been able to find that are associated with tissues in our collection. And then, 3,000 and 6,000 citations associated with those two collections, our tissue collection and our voucher collection. But there are millions and millions of data queries and downloads that are happening from the aggregators and from our web access itself. And we have no idea what these folks are using this data for. Obviously, some of them are legitimate research purposes, but there are obviously other downloads that are happening from other sources that I have no idea what they are, where they've come from, because there's no way of tracking them. There's no way of actually going in and being able to find the references to those pieces of information. So just to summarize this first part of the talk, the integration and attribution cycles are obviously complex webs of nonlinear interactions between all of these different players that are involved in the cycles. The two cycles are obviously interrelated and of benefit to all of the members of the cycle. In order for us to improve integration, we need to strengthen the technological linkages between these data components using APIs, identifiers, and other informatics methods. And then we need to engage all of the generators and users of the data to improve the social model of making everybody collections aware and making sure that all of these products are being cited in the correct way. In order to improve attribution, we need to ensure that the data pipeline is bidirectional to ensure the advocacy and continued viability and use of all of these collections. This will, as a secondary effect, also improve the data quality and the interoperability of our data with other data sources outside of our realm. So how do we achieve this? How do we actually do this? Is it as simple as a reliable, consistent technological solution? Or is it more complex than that? Are there more things that we need to be aware of in terms of solving this? Obviously, we understand that this is not something that's going to be solved overnight. But hopefully, some of the things that we're starting to put in place will work towards a point where we will be able to do that kind of thing. So with that in mind, BCoN sponsored a needs assessment workshop in February of this year. It was a 2-day workshop held here in Lawrence at the Biodiversity Institute. There was 36 attendees with representatives from all of the different actors in the data pipeline. There were folks from aggregators, from collections, from publishers, from repositories. All of the different sort of groups were represented at the workshop. The workshop report and the agenda and presentations can be seen at the link that you see at the bottom there on the AIBS page. We've posted all of the original presentations, the agenda for the meeting, and then the subsequent report, which is what I'm going to detail coming up next. So the sort of basic take-home messages of the workshop report are that solving the integration and attribution issue is achievable, we think. It is a pressing need and of vital importance to the sustainability of collections infrastructure. A functional attribution cycle requires both technological and social solutions in terms of bioinformatics tools, as well as best practices and standards to inform the community as to how best to cite this material and make those connections. The ultimate aim is to obviously create a system where navigation of the data pathway can be effected from any access point. So essentially a distributed web of knowledge. So essentially a researcher can go into GenBank and can look up a GenBank sequence, and from there can get to all of the publications, can get to the original voucher specimen associated with that, and can get to all of the images and all of the ancillary data that goes along with that particular specimen record in order to be able to make all those connections. By the same token, somebody from a collection should be able to go in at any part of that cycle and be able to make the same kinds of inferences and connections. The technical solution obviously hinges on a series of unique identifiers for the various components of the data cycle, and I'll talk about that a little more as we go on. And obviously, the main workshop deliverable was a model for such a system. So how would we put such a system in place, and what would we need to do in order to be able to do that? So looking at those unique identifiers that we would need in order to identify all of these different pieces, there's a number of these pieces that are already in place or partially in place, but we obviously need to strengthen all of them in order to be able to make all of these linkages work. So there are cool URIs that are generated by GRBio, which is the global repository of bioinformatics biocollections, where there are cool URIs generated for both institutions and collections. Then there are GUIDs that are associated with individual collection records that are minted either in the collection management system themselves. So things like Specify and Arctos and KEMU can generate these things at the source. Or they are generated by the aggregators for those who are unable to generate them for themselves. What we also require is a GUID linking mechanism to facilitate the relationships between objects. So tissue voucher, host parasite, duplicates of herbarium specimens, those kinds of things. And then also being able to link to the products that are associated with these different records. Images, sounds, videos, sequences, all of the products that are created during research and during various other uses of the data. We then need DOIs for data sets. So data sets that are downloaded from an aggregator that may then be used in publication. And obviously those DOIs need to include all of the individual GUIDs associated with the occurrence records found in that DOI. We then also need DOI to DOI linkages to account for any changes that may be made to that list of records downstream. So a researcher downloads a bunch of data from GBIF and then potentially goes in and georeferences all of that information, or goes in and does some changes to the locality information, or whatever the case may be, based on some other piece of information that they've been able to glean. So we need to be able to link the original DOI that was downloaded from GBIF to the now changed DOI of augmented information that has now been created associated with that DOI. And then obviously that needs to be repatriated back to the collection so that that information can then be used to augment the original occurrence records. We also need ORCID identifiers for agents involved in the data cycle. So it not only allows for attribution of publication of data, but also those involved in augmenting, editing, correcting records or data sets. So those people who are going and downloading data sets from an aggregator and then potentially augmenting or editing those records, they need to have credit for that. It's a positive feedback loop. The more you can give them credit for doing that, the more likely they are to continue to do it, or more people are likely to do it in the future. And so this is the sort of diagram that we came up with to try and outline what that landscape looks like. So obviously up at the top you have your institutions up at the top that are then linked to collections, multiple collections in an institution potentially, and each of these institutions has cool URIs and collections have cool URIs associated with them to be able to identify the institution or the collection from which those records are coming. Then within those collections, you obviously have, I've labeled them as UUIDs, but they could be any kind of GUID associated with the individual occurrence records coming out of those collections, as well as DOIs associated with data sets that are coming out of those collections as well. So potentially, end users who are going directly to the collections in order to be able to download data and circumventing the whole aggregator scenario altogether. Those collections are then porting their data out or publishing their data through an IPT client to the aggregators, multiple aggregators at the moment, all with their own purposes and funding in mind. And obviously, in the aggregators, all of those records are then associated with that same UUID or GUID that was associated in the collection. And there are DOIs that potentially would be downloaded from the aggregators, data sets that would then be used by the end users. You then have this huge swath of end users that are then using the data for various purposes. Obviously, we would like to have an ORCID ID associated with each of those end users, and for those UUIDs to persist through the entire cycle, through the end users, as well as the DOIs, persisting all the way through so that they can then be tracked back to the original specimens and data that was used from those individual collections. Those end users are then potentially publishing data through publication to a publisher. And again, this is where the persistence starts falling to pieces. Those UUIDs or GUIDs and DOIs are not necessarily then flowing through to publication and making that sort of last connection, which would then make it possible for us to have that attribution cycle going back to the collections. Then you have the repositories, things like Data1, GenBank, all of those kinds of places that are then depositing data or depositing products associated with records from collections. And again, those UUIDs and DOIs should persist through to those additional products. You also have other products that are being created by end users, images, sounds, videos, all of those kinds of things that are not primarily associated with the collection, but should be. And so, again, in effect, those GUIDs and DOIs should be associated with those particular records as well in order to be able to make those connections. One of the other things that we identified was that there are obviously tertiary end users who, so potentially people who go in and use a sequence from GenBank that has been created by somebody else and publish on that sequence as part of a larger study. And again, those linkages are not being made in terms of the collections are not getting credit for, the original specimen that was used to be able to create that sequence or whatever the case may be. And so obviously we need this whole cycle of data to be able to make those inferences and make those connections back to the collections from which those specimens originally came. Obviously, those three little boxes labeled 1, 2, and 3 are indicating the GUID linkages that we need in order to be able to link tissues and vouchers and hosts and parasites and those kinds of things. In the second one, the DOI and UUID relationship, obviously within a DOI, we need to have all of the individual GUIDs associated with the individual occurrence records that persist through this DOI cycle of having an initial DOI that may then change due to augmentation or editing or whatever the case may be, and making sure that that whole cycle persists. And then also these DOI linkages of linking one DOI to another, in order to be able to ensure that all of those linkages persist all the way through the cycle. So there are a couple of other issues associated with this system. Obviously, due to the preponderance of the various GUID systems that are being used in occurrence records, if you go into the aggregators, you'll notice that there are some people using UUIDs, some people using LSIDs, some people using Darwin core triplets. No matter how much people tell you that it's fairly certain that they are unique, there is no guarantee of that uniqueness. And so there is the off chance that there may be two records that have the same GUID associated with them. And there is no linking mechanism of being able to link all these things together. So the way I see it, the system could benefit from two additions. One is a true GUID minting service, similar to what the geological community uses in IGSN and the publication community uses in ISBNs. So something akin to an IZSN, where all of these GUIDs would be minted and therefore would be assured of being unique. There's two problems associated with that. One is who would host it, and the other one is who would fund it. And that has been a problem over a long period of time that is yet to be resolved. The other scenario is one of a common aggregator data cache. There is this preponderance of aggregators around the world now where we are finding that there are variations in records across all of these aggregators due to various circumstances. And GBIF has recently proposed this common sort of aggregator data cache where there would be a single data cache that all of the aggregators would then pull their data from. And that common aggregator cache would then be used in order to run all of the data cleanup, all of the usage statistics, all of those kinds of things, and thereby significantly simplify the data pathways and the whole system of ensuring that these linkages are made between these various elements. This common data cache could essentially also function as the minting service. In terms of if you have a single data cache, you could then, fairly easily ensure that all of the GUIDs that are associated with individual records are indeed unique. One of the other problems with the system is that there are two major pathways in which people are using specimen and data records from collections. One is directly from the collections. So we as collection managers get messages from people asking for specimens or asking for data downloads from our collections, or they're accessing our local web portal and doing it for themselves. And the second is through the aggregators. A lot of people are going through the aggregators now and then downloading data from those aggregators, which then allows them to keep the GWIDs along with the records. The facilitation of this integration and attribution obviously requires the use of identifiers in both of those scenarios. One of the things that we don't typically do is share identifiers along with records that are coming directly from collections. We'll usually just send out a loan form that has, KU1657, a specimen number associated with that particular record. The provision of these GWIDs is obviously not ubiquitous when we are sharing information directly from collections, and that could potentially cause problems. So this is essentially what that diagram would look like with some of those things implemented. So there you can see the aggregator cache that is being proposed initially for GBIF, IDIGBio, VertNet, and ALA. The hope is that obviously all of the other aggregators will come on board as well, and we can truly have one single aggregator cache that is then used by all of those aggregators, which will simplify the whole process tremendously. So along with the technological problems that we foresee in this, there are obviously a lot of social issues that need to be implemented as well. All of the actors in the data pipeline obviously need to be engaged. Publishers, repositories, aggregators, collections, etc. Everybody needs to be on the same page. We need community standards and best practices to be implemented along these lines to ensure that everybody is playing the game the same way. Collections-based societies, and taxonomic discipline societies as well have a role to play in promoting this plan and ensuring that everybody is doing the same kinds of things. And obviously funding agencies like NSF and others have a role to play in terms of setting policy and standard practices for specimen and data use. Obviously NSF has its data management policy, which is a little light on actual specimen information, but hopefully that will change in the future. So how can we facilitate this whole system? What do we need the individual actors as part of this system to be able to do in order to make this whole thing work? So let's start right at the beginning with the collections. The collections obviously need to use database software that allows for the minting of these GWIDs and apply these to all records, and then promote and preserve the use of these GWIDs in publication through the data cycle, not only by porting data out to aggregators, but also when loaning, exchanging, or gifting specimens out from the individual collections. They need to promote the linkage of GWIDs for the related resources through databases and through Darwin Core Publishing, so that people can find out these linkages between tissues and vouchers and hosts and parasites, etc. They may need to make their collections more visible through joining the community of aggregators and advertising their collections and the associated research productivity. They need to continue to digitize and publish more robust, complete data sets. The collections are at the heart of specimen-based research, but currently are not able to adequately showcase the credit for this contribution through attribution metrics. The integration starts with the collections, and attribution is at the heart of collections advocacy. Databasing software, the collection management software that's being used by our community, is the primary vehicle for making specimen records available, and they have a primary role to play in any integration attribution strategy. IPT is a robust publishing tool that facilitates integration with aggregators through structured Darwin Core and is employed by most of the commonly used database platforms. So we have a fairly good handle on that part of the integration cycle. But the software obviously needs to continue to advance and keep pace with community-driven needs of digitization and provide the necessary tools to create identifier-based linkages between these data elements. We need to ease the publishing of data, offer tighter integration with data aggregators, and provide necessary attribution mechanisms where possible in order to be able to facilitate the advocacy of these collections. The aggregators and repositories, they need to preserve and promote the standardization and usage of existing record identifiers. Thankfully, most of the aggregators are involved in that activity already. They need to integrate with researchers, publishers, and other end users to promote the persistence of these identifiers going forward. They need to continue to work towards a single data cache model to facilitate standardizations of services and tools. And they need to provide reliable, standardized metrics of collections use. So not only through citations, which GBIF is starting to do now, but also usage statistics. So how is our data being used? How many downloads? How many queries? Those kinds of things. One of the other things that would really help out collections is measures of uniqueness. The aggregators could provide us as the collections with a measure of uniqueness of our collection. What do we have on our collections that nobody else has? Perfect metric that you could use to advocate for your collection and show why it's important. The data cleanup and standardization assistance, that thankfully is now starting to come to fruition, thanks to the work of Tadwig and various implementations that they have put in place through the various aggregators in order for us to be able to see how our collections data is being standardized and cleaned up. Annotations and other forms of data user interaction. So facilitate people being able to go into an aggregator and make annotations on records to be able to clean up data, fix data, edit data, augment data, whatever the case may be. And then also maybe put in place some geographic and taxonomic subscription services, like an RSS feed where a researcher can go into an aggregator and can actually sign up for a particular taxonomic group or a particular part of the world and get notifications when new specimen records of that particular group are online to facilitate their research. The provision of the integration and attribution services will obviously increase participation of various collections in the aggregators, because now there will be a benefit to joining the aggregators, and it'll also facilitate publication of more data through these new collections joining the system. The researchers and other end users. Researchers and end users need to be collections advocacy aware. Unfortunately, there are too many researchers out there who are not collections advocacy aware and do not, as standard practice, alert collections when publications have been published or GenBank sequences have been lodged or whatever the case may be. They need to reintegrate products created during the use of specimens, multimedia sequences, etc., or data, corrections, augmentations, georeferencing, etc., back into from research back into the data stream, so that it gets back to the collections and we can then use that to augment the records in our collections. They need to be incentivized to utilize unique identifiers for cited occurrence records and data sets in publication and downstream products, and that's where the publishers come in. And we need technological mechanisms to remove the social necessity of linking data and products. Publishers and funders obviously have a significant role to play here. The publishers, the research deliverable conundrum can be solved by creating integrated explicit links between research and specimens, data, and research products through the use of these identifiers. The publishers and editorial boards obviously should institute identifier-based improved formalized citation of materials examined in publications and voucher materials for sequences in GenBank. The PenSoft Alpha writing tool is obviously a great step in the right direction, but more publishers need to adopt or replicate these systems in order for us to get coverage across all of the published literature. The primary focus of this is obviously on streamlining research publication, but it has a secondary and equally important consequence for collections advocacy, visibility, and linking of data. So what are the next steps? This system can only be realized through the participation and inclusion of all the actors in the data pipeline. And the hope is that through these efforts, the various actors can be engaged and persuaded as to the merits of the system. We need to further engage with the community to refine and finalize the plan. We're obviously hoping that as many people as possible will look at the plan and comment on the plan so that we can refine it and make it better. And then we want to disseminate this plan through some sort of formal publication to the outside world. So there are a number of BCoN activities coming up in the near future that are going to sort of continue this work. The next one is the second Digital Data and Research meeting being held in Berkeley in June. I'm going to be giving a similar presentation to this at that meeting, and also have a couple of other speakers lined up. Donald Hoburn is going to be speaking from GBIF about the whole aggregator amalgamation process. and various other speakers will be there as well. We'll then have a similar system at the Spinach annual meeting in Dunedin in New Zealand in August, September. I have 3 speakers lined up in a symposium there to talk about various issues from the research side and from the aggregator side. And then BCoN obviously has a number of other activities lined up. We've just recently had a workshop on the Nagoya Protocol and CBD, looking at how that's going to affect collections. There is another one coming up looking at small collections and undergraduate education and ecological data. And then a big one that's planned towards the end of the year, looking at future digitization initiatives. Where do we go from here? How do we move on from here? So that's essentially the end of my presentation. I'd be happy to take any questions or have discussion with anybody if anybody's interested.
We do have one question in the Q&A. Do you want to open up that feature? Do you want me to read it to you?
No, I've got it here.
Okay.
How does this relate to? The short answer is I don't know. I'm not quite sure what IoT is. But I will certainly go to that YouTube link and take a look at it and see.
It's the Internet of Things.
Oh, okay. Internet of Things. I really don't know. Obviously, we would like to try and get as many of these other folks who are looking in similar directions involved, or to collaborate and see if we can get it going. Yeah.
If anyone else has questions, feel free to again use the Q&A feature, or you can also use the chat, and we can the chat feature, and we could see it there as well. There's a question in the chat box.
Yeah, I saw that from Deb. Yeah, Deb and I have had discussions about this already. It is a tricky subject. It's a difficult conundrum to solve. But hopefully with some more refinement and more discussion on the topic, we can hopefully get to a system that will potentially work.
Andy, can you maybe repeat the question?
Yeah, sure. Sorry. Deb is saying UUID tracking might be simplified through a single supplier, but data providers will then have to store it on their end. Yeah, I mean, I don't understand the technological sort of implications of what that means, but hopefully somebody from the aggregator side will have some answers to how to do that effectively. Teresa says, it seems that we have a lot in common with the library community. Has anybody looked at how libraries handle some of these issues? Teresa, yes, we had a couple of folks from the KU libraries at the needs assessment workshop, and they actually gave a presentation on how the library community has dealt with these issues in the past. They're obviously a little further along in terms of the way that they have handled some of these issues. And so they gave a very interesting presentation on the way that they look at things. And so, yeah, there are some commonalities there, and the hope is that we can continue to collaborate with them in terms of trying to solve some of these, especially the sort of ISBN kind of linkages there in terms of the same kinds of questions that they're trying to answer. Katie says, how do you envision data users or researchers can report use of specimens? To whom could they submit a list of goods, for example, the aggregator, the collection? So, Katie, the hope is that they wouldn't have to. The hope is that if we can get the technological pieces put in place, that once you have these goods associated to these records in publication or in GenBank or wherever the case may be, that those connections will be made for you. That computer mechanisms can be used to then troll and find those goods and then associate them back to the collection. So you would get a report of, which goods have been published on or which goods have been reported in GenBank or whatever the case may be. If we can get those technological pieces put in place, it essentially negates the whole social issue of researchers physically having to go and do that themselves and report it back to the collections, which is one of the big problems. You know, there are, I think there are numerous reasons why researchers are either loath or you know, forget to do it. They use specimens and the publication only comes out 6 months later. It is difficult to keep track of that stuff, and I do understand that. Catherine says, is there any attempt to provide compensation to Indigenous peoples who provide contributions to these data sets used in academic work? I'm not aware of any at the moment. I do know that Nagoya Protocol is looking at some of those issues in terms of trying to preserve traditional knowledge. And, you know, they are putting structures in place that would require folks who are using data sets that include traditional knowledge to then, you know, cite those sources. But I'm unaware of anybody who's actively doing that at the moment. Pinar says, is there anything that end users can do to help with the attribution process? Well, you know, again, if the end users were collections advocacy aware and ensured that they cited materials correctly, you know, in their publications or in, you know, whatever it is that they are creating, hopefully the technological solution will take care of the rest. And we'll make those attribution mechanisms that much easier to do. But you know, just maybe, you know, until the system is fully functional, if researchers could simply alert collections every time they publish things that cite material from a collection, or alert them to GenBank sequences that have been created that cite their material. And just making sure that they cite that material correctly so that those connections can be made. Deb says, FYI, as to uniqueness, it will not be affected. The way UUIDs themselves are generated makes the odds effectively zero. Yeah, but effectively zero is not zero. I get where you're coming from, but I don't know. I'm still a little skeptical. And the IGSN solution, Nagoya, yeah, potentially Nagoya could be the IZSN minting solution. Yeah, I think there are a number of players that could potentially take that on, but I think it's, you know, it's funding and who's willing to actually host it. That's the problem. Any other questions, comments? Okay, well, thanks everybody for signing into the webinar. If anybody has any comments or suggestions, or would like to read the report and send me some comments or suggestions, I'd be happy to entertain them. Feel free to email me and let me know. The more engagement we can get from the community in terms of you know, finalizing and fleshing out this plan, the better, before we look at publishing it into the literature, so that everybody can get hold of it.
Well, great. Thank you again, everyone, for joining. And I'm trying to remember when the next webinar will be. Give me 1 second to track that down, as I forgot to close that screen out. But I think we have one coming up, and I believe in 2 weeks. And it is so it's June 13th. It is using natural histories collection data to address national challenges. Again, you can go to the BCoN website to register for that. Thanks again, everyone, and we will see you hopefully on the 13th. Have a wonderful, wonderful couple weeks. Bye-bye.
Thanks, Diane.
Thank you.